Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the Deep Dive. This is the place where we take these really dense, information-packed sources, like the core NCERT texts, and we really try to distill them into the clearest, most actionable knowledge we can for you.
And, before we even jump in, we just have to say a massive thank you to all of our existing paid members.
Absolutely.
Your support is honestly what lets us carve out the time to create this kind of super useful, really focused content. So if you're new here and you find this helpful, and if you're in a position to, please consider subscribing.
Yeah, joining the paid membership on YouTube really does help us sustain the channel. And of course, don't forget to like the video and drop a comment. We do read all of them, and we really appreciate the feedback.
We really do.
Okay, so today, we're moving past the basic structures of the plant and diving deep into its nutritional demands. We're talking mineral nutrition, specifically tracking the key concepts from NCERT Class 11 Biology, Chapter 12.
Right. The big question is, what does life need beyond just carbon, hydrogen, and oxygen? And how on earth does the plant kingdom manage all those complex chemical needs?
So what's our mission for today?
The mission is to pull out the most fundamental nuggets you need for that advanced understanding. We need to lock down what essential actually means, get into the chemistry of deficiency symptoms, and then, this is the big one, demystify the nitrogen cycle.
Which is, it's just a miracle of biology, really.
It's incredibly energy intensive. We'll get to that.
Right, let's start at the foundation. Plants are amazing chemists, right? They're pulling all sorts of stuff from the soil. Our sources say there are over 105 discovered elements, but less than 21 are actually considered essential?
Fewer than 21. And that word essential is key because it's a really strict definition. It's not just, helpful.
Okay, so what are the rules? What makes an element truly essential?
There are 3 absolute criteria. First, the plant literally cannot complete its life cycle, so it can't grow to maturity, it can't produce seeds without that specific element.
And we test that with hydroponics.
Yeah.
Growing them in a nutrient solution.
Exactly. You just leave one element out of the solution and see what happens. If the plant fails, that element is essential. So that's rule 1.
Okay, what's number 2?
The second rule is that the element has to be directly involved in the plant's metabolism. It's not just sitting there. It might be part of an enzyme or a cell wall, something active.
And the third?
The third, and this one gets tested a lot, is that its function cannot be substituted by another element. You can't just, swap in a bit of zinc for iron and hope for the best. It won't work.
So if it meets all 3 of those, it's in the club. It's essential.
It's in the club. That's the difference between something like nitrogen, which is absolutely mandatory, and, say, sodium, which might be beneficial for some plants, but isn't essential for all of them.
Got it. So once we know what's essential, the next logical step is quantity. And that leads directly to deficiency, right?
Precisely. And this brings up a really important concept, critical concentration.
Okay, what's that?
It's the concentration of an essential element below which the plant's growth is visibly held back. It's retarded. The moment the nutrient level in the soil drops below that critical point, the element is deficient.
And that's when you start to see the symptoms, the yellowing leaves, the stunted growth.
Exactly. Chlorosis, necrosis, things that directly hit crop yield. And that's why we use fertilizers, to push those concentrations back up.
Which brings us to the 2 big categories of fertilizers and nutrients in general, macronutrients and micronutrients.
Right. It's all about quantity. Macronutrients are the ones required in large amounts. Think nitrogen, phosphorus, potassium, sulfur, the big players.
And then the micronutrients?
Those are the trace elements, needed in really small quantities, less than 10 millimoles per kilogram of dry matter. Things like copper, zinc, iron, manganese.
But even though they're needed in tiny amounts, their roles can be incredibly specific.
Oh, incredibly precise. And sometimes you can figure out what they do just by looking at where the deficiency symptoms appear first on the plant.
And this is where that idea of mobility comes in. This is super interesting. So if an element is mobile, the plant can move it around, right?
Yeah. It can scavenge it from older, lower leaves and send it up to the new, more important growth.
But what about the ones that can't be moved?
That's the crucial distinction. We have to talk about immobility, and the classic example is calcium.
Okay, calcium.
Once calcium is built into a plant structure, like the cell wall, it is locked in. It is not remodelized, period.
So if there's a shortage.
The plant can't pull calcium from the old leaves to feed the new leaves, so the deficiency symptoms for an immobile element like calcium and also sulfur show up first in the younger parts of the plant, the new leaves, the shoot tips.
That is such a practical piece of knowledge. A farmer sees yellowing on the newest leaves, they're probably dealing with an immobile element.
It's a fundamental diagnostic tool. And just to wrap up this section, let's look at one more micronutrient, manganese.
Right.
Okay.
What's its big job?
Its role is foundational to, all life on Earth. It is absolutely essential for the water-splitting reaction in photosynthesis.
The one in photosystem 2, that's what releases oxygen.
That's the one. The manganese cluster is what catalyzes that reaction, splitting water and releasing oxygen into the atmosphere. Without enough manganese, that whole process just grinds to a halt.
Wow. A tiny trace element is the ultimate bottleneck for breathing.
Pretty much.
Okay, let's pivot to the absolute star player here. Nitrogen. After C, H, and O, it's the most common element in living things. It's in our proteins, our DNA, our RNA.
It's everywhere. But here's the great irony. The atmosphere is 78% nitrogen gas. It's 20 NO2. It's all around us.
But plants can't use it.
They can't touch it.
Yeah.
That triple bond between the two nitrogen atoms is one of the strongest in nature, making the molecule almost completely inert. It has to be fixed, converted into a usable form before the plant can do anything with it.
And that usable form, the starting point for assimilation, is ammonia. Text 83.
That's the goal. And getting there involves the whole nitrogen cycle. Well, we should probably clarify the part that happens in the soil first, which is nitrification.
Okay, so what's happening there?
In the soil, ammonia is first oxidized to nitrate, which is text NO210. This is done by specialized bacteria, mostly Nitrosomonas or Nitrococcus.
And these guys are chemoautotrophs, right?
They are, and that just means they get their energy not from sunlight, but from chemical reactions. In this case, they get energy by oxidizing ammonia.
Okay, so they turn ammonia into nitrite. Then what?
The nitrite is then immediately oxidized further into nitrate, text NO3, by another bacterium called Nitrobacter. And that nitrate is the main form of nitrogen that most plants absorb through their roots.
So the plant takes up the nitrate, it goes into the xylem, but it doesn't just stay as a simple ion, does it?
No, and that's a really important nuance. While the roots absorb nitrate, it gets transported up to the leaves, where it's immediately reduced back to ammonia, and then boom, it's built into amino acids.
This is converted to an organic form really quickly.
Very quickly. In fact, a lot of the nitrogen traveling up the xylem is already in the organic form as amino acids. It's just more efficient for the plant to transport it that way.
Interesting. Now let's get to the real marvel. Biological nitrogen fixation, actually breaking that atmospheric X-N2 bond.
Right, and this superpower is exclusive to prokaryotes. Only certain bacteria and cyanobacteria can do this.
The text N2 fixers.
Yep. And you have different types. There are free-living ones in the soil, like aerobic azotobacter and anaerobic rhodospirillum. And then you have the really important ones, the cyanobacteria, like Anabaena and Nostoc.
And Nostoc is a name to remember, right? It comes up in the source material as being used as a biofertilizer.
It's used explicitly as a biofertilizer. It's a powerhouse because it can photosynthesize, make its own food from sunlight, and fix its own nitrogen from the air. It's completely self-sufficient.
But doing that, fixing nitrogen, is just incredibly difficult from a chemical standpoint.
It's unbelievably demanding. The enzyme that does the work is called Nitrogenase, and the energy cost is staggering. It requires 8 ATP for every single molecule of ammonia it produces.
8 ATP. That's a huge biological price to pay. Why so much?
You're breaking one of the strongest bonds in nature, that text N2 triple bond. The nitrogenase enzyme has to pump a massive amount of energy in to tear that apart and add hydrogens to make ammonia.
And all that energy comes from the plant's respiration.
It comes from the host cell's respiration. It's a constant, heavy energy drain. And this high cost leads to another huge problem.
The oxygen paradox.
The oxygen paradox. The nitrogenase enzyme, the one doing all this expensive work, is extremely sensitive to oxygen. Oxygen literally destroys it.
So how does the plant solve that?
Yeah.
Especially in a root nodule that's actively respiring.
In those symbiotic nodules, like on legume roots, the plant has evolved a perfect solution. It produces a special pigment. It's called Leghaemoglobin.
Leghaemoglobin sounds like our hemoglobin.
It's very similar. It's an oxygen scavenger. It floats around in the nodule and binds to any free oxygen, mopping it up and keeping the environment strictly anaerobic right around the nitrogenase enzyme. It's a beautiful piece of coevolution.
A perfect little protective bubble. This all connects back to the bigger systems, right? This huge energy cost, the need to protect the enzyme, it puts enormous pressure on the plant to be efficient.
It absolutely does. And that's why we see such a clear difference in the transport systems. Xylem, which moves water and simple minerals, is fundamentally unidirectional,
Just up from the roots to the rest of the plant.
a one-way street. But the phloem, which carries the really valuable stuff, the sugars from photosynthesis and those expensive organic nitrogen compounds,
That's bidirectional.
it has to be. Its flow is dictated entirely by the source-sink relationship. A leaf-making sugar is a source. A growing root is a sink. But a developing flower bud at the top of the plant is also a sink, so the phloem has to be able to move resources both up and down.
To shuttle those precious energy-intensive compounds wherever they're needed most.
Exactly. It's all about resource allocation and energy conservation.
You know, speaking of controlling resources, there's that interesting little detail in the sources about cold storage for fruit.
Yes, the post-harvest technology nugget. It's a great example of applied metabolic control.
So we know putting fruit in a cold chamber slows down its metabolism, its respiration.
Right, lower temperature, slower reactions. But that's not the only trick. Those chambers often have an increased concentration of carbon dioxide.
Why is that?
The higher Ch12 level acts as another brake, a further inhibitor on the respiratory process. It slows down the breakdown of sugars and organic compounds even more, which delays spoilage and massively extends the shelf life.
So you're manipulating the plant's own metabolic gas exchange to preserve it. That's clever.
It's all connected.
Okay, let's try to synthesize this. What are the absolute core takeaways from this deep dive?
I think there are three main things. First, know that the definition of essentiality is strict. Those three criteria are non-negotiable. Second, you have to understand mobility. An element like calcium is immobile, so its deficiency shows up in new tissue. That's a huge clue.
And the third?
The third is the sheer cost and complexity of the nitrogen cycle. Remember that number? 8 ATP for 1 molecule of ammonia. That massive energy investment explains so much, including why the plant goes to such lengths to protect the reaction with Leghaemoglobin.
Fantastic. You know, that 8 ATP cost leaves me with a final thought to chew on.
Go for it.
If biological nitrogen fixation is one of the single most expensive metabolic processes in the entire plant kingdom, and evolution always favors efficiency, couldn't the ultimate goal of plant physiology be to achieve a near-perfect, closed-loop system for recycling nitrogen? A system where, once the plant has its initial supply, it almost never has to pay that 8 ATP toll again. What would that even look like?
That's a great question. What kind of innovations would that require?
Something to think about. A huge thank you again to all our existing members for making this possible. If you found this deep dive useful, and if you can, please do like the video, share a comment, and consider subscribing and joining our paid membership on YouTube.
We appreciate it more than you know. We'll catch you on the next deep dive.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.